All right. So I don't know what happened in class today. I heard a rumor of remediation or something like that for the exam. But I'm planning on just moving forward with the applied immunology stuff. Um, so last time I left off after talking about FACS and flow cytometry. So flow cytometry is basically a big machine that analyzes your cells one at a time. And first you have to bind fluorescent antibodies specific for cell surface antigens to your cells. In this case, the researchers are looking at CD4 and CD8. And then the flow cytometry thing is going to categorize your cells and count them in terms of their expression. So for example, these are your CD8 positive cells. They don't express CD4. And then these here are your CD4 positive cells, and they don't express CD8. So these are your cytotoxic T cells, these are your T helper cells. And then everything else is right here. They don't express anything, either of those proteins. So these are your non-T cells right here. So flow cytometry is what gives you this nice graph looking thing that counts your cells. But FACS stands for fluorescence activated cell sorter. And FACS takes the information from a flow cytometry and turns it into an ability to sort cells into different vials by giving cells an electronic charge and then pulling them with magnets into a specific tube as they fall. So FACS uses flow cytometry information to sort your cells into, for example, CD8 positives, CD4 positives, and double negative cells. That would be a FACS. This output here is a flow cytometry output, though. Flow cytometry is the more visual or graphic application of, just the counting. FACS is the actual cell sorting ability. Then we have the optiplex bead assay, which was invented somewhat recently. So optiplex bead assay is usually used to look for a specific antibody in a sample. And what it consists of is you have beads of different sizes. For example, 2 micron, 4 micron, and 8 micron beads. And the beads are coated with antigens of different kinds. So each bead of a different size has a different antigen. So what you're going to do is add your primary antibody test sample. You don't know if it contains the antibody or not, but you're going to add it to the beads. Okay. So you can see this little spike here. It has specific antibody that's pink. And then these pluses here have antibodies that are more square shaped. And then there weren't any anti-smiley face antibodies in the mixture. Okay. After you add your primary test antibodies, you add mouse anti-human IgG pre-conjugated to a fluorescent dye. And then you run it through a special FACS machine, and the FACS machine plots them on a graph of fluorescent versus size. So you can use the FACS to separate the beads into different tubes based on size and fluorescence. One application of the optiplex bead assay is a diagnostic test for Lyme disease, which is caused by Borrelia burgdorferi. If you have had Lyme disease for at least a week or so, you will have circulating antibody that is specific for a multitude of different Borrelia burgdorferi surface antigens, right? Because the immune system has produced antibodies against the antigens it's being attacked by. The beads can be used for IgM and IgG, and the presence of either class of antibody is a positive test result. So it's abnormal to have even IgG against Lyme disease. Then there's a secondary antibody. There's a sample that you add to the beads. And I believe the IgM beads are a different size than the IgG beads. So they're going to be sorted into different parts on the graph. Here's what the data might look like for that first picture. And this is what you could expect to see for the Borrelia burgdorferi as well. So your 2 micrometer population fluoresced, which means there were antibodies to the triangle, right? The spikes. And since it's really far on the x-axis, toward the left, that means it's your smallest bead size, 2 micrometers. We also had a 4 micrometer population, and that would correspond to this one here. And the reason it's fluorescing is because the sample that you tested had primary antibodies directed against the plus antigen. Okay. And then we also had the 8 micrometer population. There were no antibodies against the smiley face that was on the 8 micrometer beads. So that one didn't fluoresce, but it was located down here. So what this can tell you is which antibodies you have in your solution in a different way than just conjugating them to a bunch of different dyes. Instead, you can use different sized beads. These beads are usually made of sephrose or agarose or something inert that is like gelatinous. That's just a fun fact for you, I guess. Okay, and then a related concept to that is affinity purification. So, in this case, you have a serum sample. You have a serum sample and you want to use antibodies from that serum sample to do something. So what you want to do is fetch the antibodies that you want out of the mixture of a bunch of different antibodies. In this case, we're after the blue antibodies that you can see here. So what we do is we take an antigen of interest. For example, they might do this to produce an antitoxin against rattlesnake venom. So they're going to take rattlesnake venom antigens and they're going to bind those to a sephrose bead inside a column that looks something like this. Then they're going to add the serum from the horse that they immunized with the rattlesnake venom. Then they're going to wash everything else out, and through the washing step, specific antibody will remain bound to the beads if the specific antibody is present, which, you know, there's a good chance it's present. Otherwise, you wouldn't probably be doing this test. Then you're going to throw out everything that you washed, all of your excess antibodies and proteins and cell lysate materials, right? So what you're left with is only your specific antibody left inside the tube, inside the column. Now you should notice that these antibodies are not all going to be identical, okay? Even though they're all depicted as being identical, they're not all going to be identical. Because what you have is a polyclonal mixture of antibodies, all of which can bind to different epitopes on the antigen. Okay? Sure, some of them might be identical, and they're all specific for the antigen that you're looking for, or that you want antibodies to, but they're all going to bind to different epitopes on that antigen. Okay? So these are polyclonal antibodies still. It's just now they've been affinity purified, so you know that these antibodies that are in here only bind to your antigen. Okay, so once you have your affinity purified antibodies left in the flask or in the column, you're going to elute them out using glycine buffer, which is just a solution with glycine, and it's at a pH of about 2.5. So that disrupts antigen-antibody binding and allows you to get the antibodies back. Now what this can be used for, now that you have all these antibodies that are specific for rattlesnake venom, you can infuse these into a patient to treat them for a rattlesnake bite. That's just one of the options. First, you're going to have to neutralize your solution by just putting it in saline or tris PBS or something. But you now have a collection of antibodies that are useful because they bind to an antigen of interest. For example, rattlesnake venom proteins. So that is affinity purification. And this is Janeway's depiction of it. Antibody to antigen A might be bound to beads. So this is when you want to do it the other way around. You can actually pull an antigen out of solution by using antibodies bound to beads in a column. So you have antibodies bound to beads in a column. They're specific for the red antigen in a mixture of molecules. For example, maybe you're interested in a nuclear protein, and so you make antibodies to it, but you want to pull those antibodies out of the serum of the mouse that you immunized, right? Everything else in that mouse's serum isn't necessarily going to be specific for your antigen. So the nuclear antigen is in red here, and only the nuclear antigen is going to bind to the antibodies. And then you're going to wash away all of the unbound molecules by using tris-PBS. And now you're left with only the antigen of interest in the mixture. Okay, so you can also use this to fish an antigen out of a heterogeneous solution. And then at the very end, you can elute the molecules by using glycine buffer, just like you can use with any antigen-antibody interaction. So you can use it to fish for antibodies, you can use it to fish for antigen, it just depends on what you have bound to your bead. Okay. Another technique is called immunodouble diffusion. More commonly, this is referred to precipitin reaction. The precipitin reaction, okay? This is one of the earlier immunotechniques. So the way that this works is you have antibodies in a well, and you're looking at an agarose gel here, an agarose gel, okay? You have antibodies in a little hole or a little well in the agarose gel, and then you have antigens in some neighboring wells that are a couple of inches away. Now all that you do is you add your antibody, you add your antigens, and then you just wait a couple of hours, and the antibodies are going to diffuse through the gel just by diffusion, and the antigens are going to do the same thing. They're going to diffuse from their wells, and they're going to diffuse radially outward, just like the antibodies are. And then wherever an antibody meets an antigen, it's going to solution, or it's going to form a precipitation reaction where you can see a clear band. So in this case, we have an antibody against it's an anti-1 antibody, so it's specific for antigen 1. Okay, so anywhere it encounters antigen 1, it's going to form a precipitation ring, or whatever you want to call this. So there's antigen 1 here, and there's antigen 1 here. So it's going to encounter antigen 1 in both those places, right? So here's what you see. Now what you're going to have to do in order to actually see this precipitation, you have to stain the agarose gel with Kamasi blue dye, which binds to proteins and makes them evident. It's just a blue color. And then you'll have to destain, and then you'll be able to see it through a clear gel. In the second case here, what we have is a polyclonal antibody mixture where there are antibodies against antigen 1, 2, and 4. And then in our wells, over here we have antigen 1, and over here we have antigens 2 and 4. Okay? So where the anti-1 antibody meets the 1 antigen, it forms a precipitation. Where it meets the 2 antigen, it forms a precipitation. And where it meets the 4 antigen, it forms a precipitation. Okay, so the anti-1 antibodies are here, anti-2, and anti-4 are here. Okay, so that's why you see multiple precipitations. So this can allow you to look at not only one antigen to determine if you have antibody against it, you can look at multiple different antigens to see if you have antibodies against all of those antigens in the same reaction. Lastly, in our third reaction here, we have antibodies against antigens 1 and 2. Okay. So we have antigen 1 in this well, and we have antigens 1 and 2 in this well. Anywhere antigen 1 is found, the antibodies against antigen 1 are going to bind. And anywhere antigen 2 is found, the antibodies against antigen 2 will also bind. So you can see that these antigens are sort of the same size here, and that's why you have this little spur coming off of it, is because that is anti-2 antibodies and antigen 2 coming together and precipitating. Okay, you don't see the spur on the other side because there's no antigen 2 over here coming from this well, right? So here's what this actually looks like. You start with it looking like this. You have antigen 1 and antigen 2 in 2 different wells, and then you have your antibodies, okay? Now, an antibody meets antigen, there is a precipitate or precipitate that you can see after you stain the gel with Kamasi blue dye. So as time passes, these molecules diffuse outward radially, like the circle that they're in enlarges and enlarges and enlarges. Now eventually those circles begin to overlap, and wherever those circles begin to overlap, you have antigen-meeting antibody, and you begin to form, basically you begin to form the precipitate, right? So this is antigen 1 binding to anti-1 antibodies, and then this is antigen 2 binding to anti-2 antibodies. As time progresses, as more time passes, these circles continue to expand until they're, pretty much completely overlapped. The precipitate grows to look more like this. And so you have, you still have the same precipitate occurring. It's just now more antibodies have come to encounter their antigen because more antigens and antibodies have diffused outward. Okay, so this one takes a little bit of a while, probably maybe a day, anywhere from a few hours to a day. But at the end of it, you're going to have to use Kamasi blue to stain these proteins to make this reaction actually visible to yourself. So that's how the precipitin reaction works. I'm keeping my ear out for any questions, just so you know. So, one use of this is that you can make an antibody like in a mouse or something, but you may not always know what class of antibody the antibody is. So maybe you want to make an IgG molecule, right? That's usually the most useful class of antibody to create. It's sort of the most efficient class. It's very tightly binding, and it doesn't cause any kind of allergic reaction. It just simply opsonizes the target, and can also neutralize the target, right? And it has good tissue penetration, so IgG is usually what you want to make. Now, if you're making an antibody using mice, you probably don't want to make IgE molecules, because that could cause the patient to have an allergic response to the antigen, right? You probably want to make IgG, or potentially you might want to make IgM. Usually IgG is what you're after. So what you can do is you can put anti-mouse IgG in the left well. You can put anti-mouse goat anti-mouse IgE in the right well, meaning you have molecules, you have IgG molecules made by a goat that are specific for mouse IgG FC regions, and you put those in the left well. Yes, you can buy that online. And then in the right well, you have molecules of IgG made by a goat that bind to mouse IgE antibody FC regions. So that's goat anti-mouse IgE. Refer to slide 2 if you're confused about that terminology. Goat anti-mouse IgE is in your right well. And then you just run a precipitin reaction and you see where the line results. If the antibody you made is IgG, you're going to see a line forming between the IgG molecules that you made and the goat anti-mouse IgG as those proteins come into contact. Okay, so this allows you to positively identify that you have made an IgE class antibody, and it's relatively easy, simple, and cheap. And it doesn't take a super long time. You don't have to do any kind of protein sequencing, nothing like that. You can simply just run this reaction and have good faith that it worked. Now the next one is, go ahead. I hear a question.
Can you explain the spur again? And I think it was another slide. Yeah, that one.
So the spur is antigen 2 meeting anti-2 antibodies. So basically antibodies from here have diffused in this direction, and they're specific for epitopes of antigen 2, right? So they skipped past antigen 1 until they finally met their fate with antigen 2 right here. You can also sort of see spurs here, here, and here because these antibodies have also passed through the antigen that they're not specific for and then finally met their antigen.
So in number 2, the non-identity, there's also spurs.
Yes.
Okay. All right. I was trying to compare the two.
Mm-hmm. Okay, so our next one is QuantiFERON-Gold. Some background for this one. It's not as hard as it probably seems. This one is basically used to diagnose tuberculosis or historical exposure to tuberculosis. Tuberculosis, Mycobacterium tuberculosis, also abbreviated MTB, is the causative agent of the lung disease tuberculosis, or TB. I'm sure everyone's heard of tuberculosis, right? This is the most deadly infectious disease in the entire world each year, other than COVID now. So it's a pathogen of the respiratory tract, and a third of the world's population has been exposed to tuberculosis and has been infected with it. The infectious dose for tuberculosis is like literally 1 cell. So this one's a pathogen of the respiratory tract. And the thing about it is 90% of the people exposed to tuberculosis enter latent infection. Okay, so they don't have acute symptoms or anything like that. Their immune system actually forms a granuloma around it, around the bacterium in the lungs, and just sort of walls it off so it can't grow. That response is usually effective, but sometimes it's not. In any case, we need to screen all healthcare personnel for tuberculosis to make sure they don't bring it to patients, right? Now, one of the issues with tuberculosis is that if you got the BCG vaccine for tuberculosis, and you do a traditional skin test for tuberculosis, your skin test will come back positive because you had an immune response against the BCG vaccine contents, right? The BCG vaccine is basically an attenuated vaccine for tuberculosis. I think it uses a different mycobacterium species that's very similar to tuberculosis but doesn't cause severe disease. The BCG vaccine is given to patients, it's given to people who are at high risk of developing tuberculosis. Tuberculosis predominantly affects people in low socioeconomic statuses. It's pretty well controlled in the United States, but we still have, about 10,000 cases of tuberculosis a year. So definitely a lot of improvement to be made. So the BCG vaccine, some people have received it if they are at high risk, and it's especially given to children because it's especially effective in children. So how the QuantiFERON-Gold test gets around this traditional skin test being positive, here's how the test works. You draw blood from the patient to be tested, and you add a couple of things to it. There are 2 tuberculosis peptides inside the vial. ESAT6, CFP10, both of those are tuberculosis proteins. Okay. You also add peanut agglutinin. Sometimes that is abbreviated PNA or PHA. PHA, this is a mitogen. What that means is it is a molecule that stimulates cells to grow. It stimulates them to proliferate. And it serves as a positive control and can adjust for any background signal in this test. ESAT6 and CFP10, those are proteins, those are peptides, and they're going to be taken up by monocytes in the blood vial, and then they're going to be, they're going to be phagocytized, shredded into pieces, and loaded onto MHC2, and then they will be presented to CD4 positive T cells in that vial. Now, if any memory Th1 cells are present, they're going to make interferon gamma in very high amounts when they're exposed to those epitopes, right? Because they're specific for the tuberculosis peptides, because you have been exposed to tuberculosis, therefore you have memory Th1 cells to tuberculosis. So, what the QuantiFERON-Gold really is a sandwich ELISA. It's a sandwich or a capture ELISA. And it uses antibodies that are specific for interferon gamma. So it's looking for interferon gamma. The benefits of this is that, unlike the tuberculin skin test, with QuantiFERON-Gold you only have to make a single doctor appointment. you don't have to come back to get your results read because the machine does all the work for you. Usually when you get tested for tuberculosis, they inject you with a little tiny, they basically inject you with tuberculin, which is a protein from the bacterium, and they inject it under your skin, okay? Now, after that, they wait 2 days and you come back and get your skin test read, or 2 to 3 days, and then they measure how much inflammation you have, you know, how large the red circle becomes, basically. They measure it in millimeters from the injection site. So how much inflammation resulted. Anything above like 5 millimeters is seen as a positive result. So that indicates that you had an inflammatory response against the tuberculin protein. That means you've been exposed to tuberculosis. So you need your chest x-ray done to see if you have active TB, or to rule out active TB and decide that you have latent TB if you don't have a positive x-ray. So that's how the tuberculin skin test normally works. One of the issues is that you have to come back and actually get your skin read by like a nurse. Um, the second benefit of QuantiFERON-Gold, besides just allowing you to only make one appointment, the second benefit is that it is unreactive for the Bacillus calmette-guerin BCG vaccine that uses a weakened version of Mycobacterium bovis. So that is a vaccine that is given to high-risk individuals when they're young so that they don't develop tuberculosis. It's not as effective in adults, and they don't even bother because it's really not very effective in adults. So it's only restricted to children. So if you receive that vaccine, the QuantiFERON-Gold doesn't give a positive reaction, unlike the skin test. So this is actually the only option that people have if they've received that vaccine. There are other assays that get around the vaccine thing. There's an ELISA assay that they can use, but this is one of the first non-traditional tuberculosis diagnostic tests. So when you do the QuantiFERON-Gold, you have 4 tubes. You have a purple tube that contains a mitogen that's going to stimulate cell proliferation. And it's a positive control, so you're going to get interferon gamma production in that vial because everybody has, you know, memory Th1 cells in their blood. Then you have a gray tube that is the negative control. It contains nothing, and it adjusts for background interferon gamma. So this is kind of what you subtract away from your signal. And then you have the green tube. It detects CD4 T cell responses, and then you also have a yellow tube that detects CD4 and CD8 T cell responses. That has to do with what peptides are added to those 2 vials. So here's sort of how it works. This is Sanders' figure. I actually don't really like this figure. But anyway, so whole blood is added into 4 wells of a 24 well culture plate. 3 drops of the antigen are added to each well. The plate is shaked for 1 to 2 minutes and then incubated for 16 to 24 hours. In that time, interferon gamma may be produced, depending on whether the person has been exposed to tuberculosis or not. If they have been exposed, interferon gamma is going to be produced within 16 hours by the memory Th1 cells. Remember, they're very rapid responders. So what you do after you shake and incubate is you harvest, you suck up the supernatant from the plasma in each well, and then you store that supernatant in racked microtubes or uncoated microplates. And then you add it to an interferon gamma sandwich ELISA. Now, on this plate, you have antibodies against interferon gamma. You have antibodies coated on the plate that are specific for interferon gamma. So their FAB region is facing up. Their FC region is literally conjugated to the plastic by a covalent bond. You add the supernatant from your test, from your sample, into each well, and then you wash. to wash out all of the excess interferon gamma that hasn't bound to antibody. Then you add a secondary antibody. Your secondary antibody is specific for interferon gamma, but it's specific for a distinct epitope on interferon gamma. So it's going to bind to a different location on the antigen than the primary antibody. Okay? Then you wash out any excess secondary antibody. You wash the plate, prolifically, and then you add the substrate, the chromogen substrate, and you wait 30 minutes. Any secondary antibody that is left over that has bound to antigen, it has an enzyme that is going to convert the substrate into a colored chromogen product. And that literally produces a color that is read by a computer. Any color production, usually it's like blue or red, is the color that is produced when you add the substrate. The computer is going to read whether there was any color produced, and it will actually calculate the results using a certain software that the developers of this test have made. So it has like cutoff points for interferon gamma production, and it has been verified like thousands and thousands of times at this point to be very accurate for detecting people who've had prior exposure to TB. Now, if you test positive for TB by QuantiFERON-Gold, you still have to go get a chest x-ray to see if you have active tuberculosis. Your chest x-ray is either going to look, white because you have a bunch of inflammation going on, or it's not going to look like it at all. It's going to look like a healthy lung. In that case, you have latent tuberculosis. So you need the chest x-ray to differentiate between whether you have acute or latent tuberculosis. In most people, they're going to find that they have latent TB, if they have it at all. Only about 5% of people develop acute disease with tuberculosis. Okay. So I don't hear any questions. So it's a sandwich ELISA for interferon gamma that is generated by memory Th1 cells in whole blood. And then the next concept is something related to vaccine immunology, the Hapten-carrier concept. Now there's two words here. There's the hapten and then there's the carrier. A hapten is an antigen that is not immunogenic on its own. What that means is it doesn't generate an immune response on its own. A hapten alone cannot cause an immune response because it can't activate T cells via MHC2. So usually with haptens, you're looking at things like polysaccharide capsules, right? So for example, we're looking at a capsule polysaccharide here, like in the one in Streptococcus pneumoniae, which used to cause tons and tons of cases of meningitis in babies, but nowadays doesn't really cause any meningitis in babies because there is a conjugated vaccine for it. So what they do is they know that the hapten is a problem because it can't generate a strong immune response on its own. So they take the hapten, the polysaccharide, and they link it to a protein. They can use the diphtheria toxoid, which is an inactivated toxin made by the diphtheria-producing bacterium. They can use a number of different toxoids. A toxoid is just a toxin that's been inactivated, usually with formaldehyde. And they conjugate those two things together. Now, the polysaccharide, there's going to be B cells that are specific for the polysaccharide, right? But normally, if you just immunized people with the polysaccharide, they couldn't get T cell help in response to that because there's no protein there. So they took the protein, they added it onto the polysaccharide, and the protein contains T cell epitopes that the B cell is going to take up along with this whole complex, and then it's going to shred the protein and the carbohydrate into little tiny pieces, and it's going to load all the peptides into MHC2. And it's going to present those MHC2 molecules and that T cell epitope to a T cell, a T helper cell. And that T helper cell is going to provide the cytokines that allow the B cell to undergo class switch, determined to turn into a memory B cell, to undergo somatic hypermutation, to become long-lived memory B cells. All of those things that B cells do that we're so interested in making vaccines for, yeah, they all depend on T cell help. Okay, so you need this protein in order to generate a response against a polysaccharide. So the carrier is a non-self foreign protein molecule, typically a toxoid, and it's been conjugated to the hapten. And what it does is it confers immunogenicity. So it allows an immune response to actually happen, a memory immune response. So it affords T cell help to the B cell. Therefore, you have the Hapten-carrier concept. You want an antibody response against the hapten, the polysaccharide, so you add a carrier molecule, which is the toxoid, and that allows the B cell to receive T cell help in the form of cytokines. And that drives the B cell to make anti-capsule antibodies and to turn into a memory B cell and to undergo somatic hypermutation and all that fun stuff. This vaccine can be given to children as young as 6 months old, and that is actually really important because children 6 months old are at extremely high risk for Streptococcus pneumoniae infection, which can lead to meningitis and can be deadly. And it almost invariably is deadly if you develop meningitis in response to Streptococcus pneumoniae. The Haemophilus influenzae type B vaccine is another example of a Hapten-carrier vaccine, or conjugated vaccine is what these are called. That one also causes a lot of meningitis in babies. They've reduced the rate of disease due to Haemophilus influenza type B by 99% or more. It used to cause 200,000 deaths per year, but now it causes less than 100 deaths per year in infants. So it literally saves hundreds and hundreds of thousands of lives a year. And that's with only that disease. So you can imagine when this is applied to like everything that the human race used to suffer from, it's really reduced a lot of death and disease. So that is the Hapten-carrier concept of conjugate vaccines. Hopefully I explained it to your liking. And then we have some ELISAs. And ELISAs are relatively simple immunoassays that you may have done in like cell bio or another class. The first kind of ELISA is the simplest and is the direct ELISA. ELISA actually stands for end-linked immunosorbent assay. So it's an immunoassay that has an enzyme on the end of the antibody molecule. So the direct ELISA is typically used to detect specific antibody in a serum sample. However, it can be used to detect antigen. Okay? It's usually used to detect antibody, but it technically can be used to detect antigen. So, what happens is this. You start with a well in a plastic plate like this, and in each of those wells, now usually there's 96 wells, in each of the wells you have antigen molecules coated on the bottom of the plate and up on the sides as well, typically. They are bound to the plate by a covalent bond. Okay. You add a test serum to that liquid that antigen is bathed in. So usually you're looking at tris PBS is what this will be incubated in, because tris PBS is just a really inert solution that proteins are happy in. So you add antibodies into your tris PBS and you are looking for an anti-antigen antibody, right? So in this case, you're looking for antigen-specific antibodies that are specific for your green antigen you've coated on the plate. So the orange antibody then, because it's specific for the antigen, it's going to bind to the antigen, and it's going to bind really tightly. The green antibody is not specific to the antigen, and it does not bind to the antigen to any appreciable extent. Next, you wash with a bunch of treatments of Tris PBS to just rinse all of the green antibodies out, all of the non-specific antibodies. Those come out of solution. So they are discarded. That's basically all of the antibodies that you don't care about that aren't specific to your antigen. And what you have left in the flask at that point is only orange antibodies, right, that are specific for your antigen. Next, you're going to add the substrate for the enzyme that is located on the end of this antibody. Any antibodies still present, they have the enzyme, and the enzyme is going to convert the substrate into a colored chromogen product. So you can see the solution turns orange, or it might turn red or blue, depending on what you're working with. And you can actually quantitate the color change and compare it to a standard ladder or a standard curve, and that can allow you to measure the concentration of the antibody as well. Usually you have a molar excess of antigen. You don't have as much antibody as antigen. So you can measure the concentration of the antibody by comparing this to a known standard curve. A typical enzyme that they use is horseradish peroxidase. That's probably the most common one. And that one can oxidize really, really weird sounding organic compounds into things that are colorful. Usually they have really long, confusing names. I heard a question.
So what is the point of the green antibodies then? Is it just kind of like to wash out anything that's unwanted in there?
So the green and the orange antibodies both came from like a human or a mouse or something. You're only interested in the orange one though. But as we know, you have a whole bunch of different antibodies floating around in your serum, most of which are probably not specific for the pathogen.
Okay,
So we don't care about those ones.
Right.
Yeah. So those are just other antibodies that we don't care about in a heterogeneous mixture. Okay. So that is the direct ELISA. Here's another slide showing you the direct ELISA. If you don't like that previous view, I think this is a pretty simple way of depicting it. But Janeway, of course, has a more complicated way of doing things. Janeway indicates to us that we can measure the absorbance of light by the colored product, and that can actually quantify how much of the antibody we have in the sample based on known standards. And then this is from, I don't even know what book this is from. I pulled it from Sanders' PowerPoint. The construction of a standard curve where you plot absorbance versus known concentration can allow for the quantitation of the amount of antibody in a sample. In addition, one way to measure antibody titers is what they're called, which is just the amount of antibody in someone's blood, is by doing an ELISA until you don't get a positive result. So you're going to dilute it by a factor of 2 a bunch of times, and they're going to do it, 1 to 2, 1 to 4, 1 to 8, 1 to 16, 1 to 32. So these are getting less and less concentrated as you go. Now this person right here, his or her last positive result appears to have been at the 1 to 64 concentration. So we would say that their antibody titer is 1 to 64 for that pathogen. This person had their last positive result probably around 1 to 4, so their antibody titer is 1 to 4. The higher the number is, the more antibody there is in the sample. Okay, and then they also always run a positive and negative control just to make sure that the test is running, effectively. Again, you're going to use horseradish peroxidase, and then you're just going to add one of its substrates to each well and see if the enzyme is present, and if it's present, it will convert that substrate into a colored product that you can measure the amount of color change for using like a plate reader or something like that. Now, you might have done this in cell, or possibly not. So that was the direct ELISA. That only involved one antibody. Okay, it only involved one antibody that had an enzyme on it. There's also the indirect ELISA, and in this case, you have a secondary antibody. This one's actually more common than the direct because not everyone wants to spend their days conjugating enzymes to antibodies. So in the indirect ELISA, you conjugate your antigen to the plate. So you coat your plate with antigen. Then you add your antibody sample and you wash out any excess antibodies that are not bound to the antigen. So all of the non-target antibodies that you don't care about are washed out. Anything else that's floating around in the serum that you don't care about is washed out. Once your antibody is bound to the antigen, once your primary antibody is bound to the antigen, you add your secondary antibody, and the secondary antibody is conjugated to an enzyme. The secondary antibody is specific for the Fc region of IgG. Okay, so this might be mouse anti-human IgG, for example, if you're testing for a human antibody. So the mouse anti-human IgG is going to stick to the Fc region of the IgG molecule from your patient, and then you're going to wash any antibodies that have not stuck out. All of the antibodies will be washed out that are not bound to something. Then you're going to add your substrate, and if the enzyme is present and therefore antibody is present, both primary and secondary, there will be a color change. Okay, so the only added step in the indirect ELISA is that you use a secondary antibody that is specific for the primary antibody, and the secondary antibody is the one that carries the enzyme that converts the substrate into a colored chromogen product. Okay. So color production indicates your antibody was present in the test serum, and it's a positive result. So a reminder here that secondary antibodies only bind to IgG molecules that are within an immune complex, meaning that they're bound to antigen. So in other words, pathogen-specific antigen must be present in the sample for the secondary antibodies to bind. Okay? Now this should say pathogen-specific antibody. Okay, now this one is a lot less common because it doesn't always work that well. This is the indirect complement amplified ELISA. Basically, this is the same thing as an indirect ELISA, but the secondary antibody is specific for C3b. So remember that the IgG molecules in a serum sample can activate the classical pathway of complement. So when you add an antibody to a sample, C3b can actually activate on the surface of the antibody through the classical pathway. Potentially, if you're working with IgA, you could also activate the alternative pathway. Although IgA isn't as good at activating complement as IgG is, you could be looking at that. But what happens is the classical pathway gets turned on, and C3b molecules get deposited all over the antibody. Remember, C3b into like anything, literally anything that has a hydroxyl group. So you know that you've added a serum sample, and that serum sample likely activated complement and led to C3b depositing on your antibody. You can actually use a secondary antibody that is specific for C3b. Okay? C3b becomes coated on the antibody, the primary antibody, and then the secondary antibody binds to the C3b molecules. Then you wash everything out. You're still left with your secondary antibody in there because it's bound to the C3b. Then you add the substrate to this, and if the secondary antibody is present after the wash, meaning it has remained bound to something, i.e. C3b, then you're going to get a color change that you can measure again and become quantitative about. Now this one you can't be quite as quantitative because sometimes complement is a little fussy. It doesn't always deposit to the same amount on something. This one you probably don't want to do quantification for. But this is just another version of an indirect ELISA, basically. This one's, honestly, I think it's kind of pointless, but this is one thing that you can do. And then lastly, we have the sandwich ELISA, which I probably should have introduced to you before I talked about the QuantiFERON-Gold test, right? So this is what the QuantiFERON-Gold test uses. So in the sandwich ELISA, which is also called the capture ELISA, most people call it the sandwich ELISA, you have two known antibodies, okay? One is bound to the plate by its Fc region, so it's conjugated by a covalent bond to the plate. The secondary antibody, which is the other antibody, recognizes a different epitope on the target antigen that you're testing for. So in this case, you're testing for an antigen in the sample. Always. You're always testing for an antigen in the sample when you do a sandwich ELISA. The secondary antibody recognizes a different epitope of that antigen, and it is conjugated to an enzyme. So this orange one here would be our secondary antibody. So you suspect that the antigen is present in your sample, but you want to do this so that you can confirm that it's present. So you first conjugate your primary antibody to the plate. You wash, then you add your antigen. The antigen binds to the primary antibody. You wash. Any antigen that's in excess or that you aren't interested in gets washed out. Then you add your secondary antibody, and then you wash. Any excess secondary is going to get washed away, including any secondary that's present, like if you don't have the antigen in your sample. But if there's secondary antibodies still remaining after that wash, then when you add the substrate after that, it'll change the color of the substrate to something visible. Okay? So, QuantiFERON-Gold tests for interferon gamma, that's its antigen, using a sandwich ELISA. You have two anti-interferon gamma antibodies that you use in that test. The sandwich ELISA is often used for quantitative purposes. It's very sensitive. And then there's a little bit of a cheaper version of an ELISA that can be mass produced, and that is the enzyme immunoassay, EIA. An EIA, or an enzyme immunoassay, is kind of like a mini ELISA that you perform using a pre-made plastic test kit that has a see-through window in which one or two colored lines will appear. So either one colored line is going to appear, that's your control, or the control and a test colored line will appear at the end of the day. Okay? So what you do is you're testing usually for an antigen that is specific to some pathogen. For example, the COVID tests that are rapid antigen tests, they are EIAs. So you're looking for the COVID-19, I think it's the spike protein. It might not be though. It might be the L protein that they look for. But in any case, you add a mucus sample to an EIA, and then the EIA tells you if the mucus sample contains COVID antigens, right? SARS-CoV-2 antigens. There's also an EIA for Entamoeba histolytica, which is an intestinal pathogen, a parasite of the intestine, and can cause rather severe disease. So what happens is, whatever the sample is, you take an aqueous sample, you suspend it in a sample buffer, which is usually just saline, and then you apply that aqueous sample to an absorptive paper-like material in a sample well at one side of the kit. So your sample well is like located over here at the starting point. So this is where you add your liquid samples that you think might contain the antigen. You want to test for the antigen in the sample. Now, inside this kit, behind the plastic, behind the plastic wall that you can't see through, there is paper, basically. And the paper inside the test kit slowly becomes saturated with the aqueous sample as a liquid front advances forward. So the paper is going to start, soaking up all of that liquid, and it's going to slowly diffuse until it reaches the test window. Okay? Now, on the paper near the beginning, like around here, you have pathogen-specific antibodies, and those pathogen-specific antibodies will encounter any antigens in the sample if they're present. Any pathogen antigens that are present. Okay, usually they're looking for a specific antigen that they know is a good candidate for testing for, because it's identical in every case, or it's an abundant antigen or something like that. So, if the sample contains antigen from the pathogen, then those pathogen-specific antibodies bind, and they begin to be taken along with the liquid front because they are soluble in water, right? So they begin to be pulled into the test window. Any immune complexes in the liquid front are going to become trapped by anti-FC secondary antibodies in a test strip in the test window. So this contains anti-FC, anti-IgG secondary antibodies that are conjugated in a test strip to the paper within the test window, okay? So it's in the test window so you can actually see it. Excess antibodies, yes, there will be excess antibodies. Excess antibodies in the liquid front, they might be bound to antigen, they might not be, they're probably not. They flow past the test strip because that's going to become saturated. They flow past it and onto a second control strip where they are trapped by anti-horseradish peroxidase antibodies. So antibodies that are specific for the enzyme that is linked to the antibody. All the other molecules are then washed out of the kit with wash buffer, which is just tris PBS. They don't tell you that, but that's all it is. And then the HRP conjugated antibodies in the test window convert an added substrate into a colored chromogen product. So it's really just a mini ELISA that works with paper. And, you know, I guess it probably costs a little bit less money. You need less expertise to do it. You can do it for cheap. You can mass produce these. They're really small. You can mass produce these for like almost no money and then charge, you know, $20 for it and probably make $19 in profit, because it really is just a piece of plastic, a piece of paper, and some antibodies and some saline. I mean, really cheap stuff. So that is the EIA. I added that slide recently because Sanders talked about it. So he added it to the class. And then another variant of an ELISA is the radioimmunoassay, or the RAI. Radioimmunoassay. To make this brief, the radioimmunoassay is an ELISA where you don't use an enzyme, but instead you use a radioactive element to label your antibody. Okay? So this is just the radioactive version of the ELISA. Now this is obviously a lot more dangerous, but it's also a lot more sensitive, and you can use it for quantitation to very precise numbers. So that's kind of the utility of this, is it can be very quantitatively precise. But it's unfortunately very dangerous. You have to wear like radioactive proof stuff, which is usually like lead coatings and such. But it works the same way. You know, you bind the antigen to the plate, you add antibody, you add a labeled ligand, or you add a secondary antibody that has a radioactive element conjugated to it. And then you just measure the label. You measure how much radioactivity is, you know, counted. To count or measure the radioactivity, they actually have these big machines called like beta counters or like gamma counters. So it depends on what you're working with, what you're going to use, but they have a machine that does it for you. You won't be able to actually see the radioactivity, unfortunately. So the radioimmunoassays, RAI. You have an immunoglobulin tagged with a radioactive substance that you can detect in a machine. You will detect radioactivity if your antigen is present because your antibody will have bound to the antigen, and then you washed out any excess, and the antibody, if it's still there, will be radioactive and detectable. So it's very sensitive, so it's used for quantitation, but it's difficult and annoying because of the hazards of the radioactivity. The two most common elements that they use are iodine 125 and hydrogen 3. Iodine 125 is a lot more common. They can use pretty much anything, though, that they can detect, any radioactive isotope. If labeled antibody has bound, then radioactive signals like energy and fragments from beta and gamma decay will be given off, and you detect these using instruments like the gamma counter and the beta counter. So iodine 125 gives off gamma and beta-like energy, and hydrogen 3 gives off beta energy. There's also this weird thing where a scintillant can take beta energy and turn it into photon energy, which shimmers when exposed to beta energy. And then you can measure the light that comes off of this using a photometer or a spectrophotometer. So interesting stuff, but unfortunately, Not so useful because of the hazards, and this is almost never done now. This is like really cool stuff.